It's my privilege to introduce our work at Design to develop a highly unique approach to treat the underlying genetic cause of Fuchs' endothelial corneal dystrophy, or FECD, which is a progressive disease driven by cellular dysfunction and loss of cell density with no approved pharmaceutical that are disease-modifying. The IRIS registry estimates about 2 million cases in the United States with Fuchs, with the primary approach being watchful waiting. About 18 to 30,000 of these cases end up with a keratoplasty annually, and a majority of these cases are driven by a single gene mutation called CTG 18.1 in the literature, which is a CTG DNA expansion in the TCF4 gene. This mutation causes cellular dysfunction because the mutant gene makes a toxic RNA. We have invented a new kind of small molecule genetic medicine, which is designed to turn off the production of the toxic RNA and thereby restore cellular health. The single toxic allele is shown with a red CTG expansion, and a company called 3X Genetics, in collaboration with us, has now developed a CLIA-approved test for this genetic mutation with a simple cheek swab. The RNA polymerase reads this gene, and the C's and G's fold over on themselves. The splice proteins see these hairpins as splice junctions and get stuck in this RNA hairball, which can actually be seen by immunohistochemistry. DT168 has been designed to recognize these expanded repeat mutations in the DNA and shut off the production of the toxic gene product while leaving the normal copy alone. Recall I had said you could see these toxic RNAs, and in the above panel, we obtained what would have been discarded endothelial cells from a keratoplasty, and you could see these abnormal toxic foci as dots inside the nucleus. DT168, shown below, is able to get rid of over 90% of these foci and is a very potent molecule with the dose-response curve shown in the right with a low nanomolar IC50 value. The graph on the left shows that increasing drug concentration has no effect on normal TCF4 RNAs. And what we also see in blue is that a number of genes have poor splicing without the drug, and with increased drug concentration, the splicing of all of these genes goes back to normal, showing that getting rid of the toxic RNA can result in restored normal splicing and therefore cellular health. Remarkably, DT168 is an eye drop, and we have just completed the first in-human study with the schema shown here, where 24 healthy volunteers participated in both a single and multiple ascending dose trial. I'm pleased to report that the results show that DT168 eye drops were well tolerated in all subjects. There were no SAEs and no clinically significant findings observed in any safety assessments. And as expected, the pharmacokinetic analysis in peripheral blood demonstrated systemic exposure below limiter quantitation across all time points and groups. So what next? We're interested in seeing if 168 does what it is designed to do in the corneal endothelium of Fuchs patients. Here are new results from a reference range study we've conducted from about 24 patients with endothelial cells from both Fuchs donors and unaffected donors. And the splice events from Fuchs cells are shown in red and the unaffected in green, and you can see a measurable separation in this data. This type of biomarker has been used in other CTG repeat diseases like myotonic dystrophy, and we think these results are the first in the FECD field and potentially enable the conduct of a biomarker efficacy study. And the planned phase 2 study approach is shown here, where we're going to take TCF4 positive patients who are already scheduled for keratoplasty, will be enrolled in a phase 2 to receive 168 about a month or more before their scheduled surgery. And since the endothelial cells are to be removed anyway, it gives us a chance to measure the drug impact on improving spliceopathy in the corneal endothelium based on the reference results we've generated from affected and unaffected eyes. And the study is not designed to find clinical effects in these later stage patients. So in summary, these eye drops were well tolerated, and it supports advancement into phase 2. We further report a biomarker assay potentially suitable for clinical use, and a planned phase 2 study slated to begin in the second half of this year with results expected next year. And we think that DT168 could be an exciting new genetic medicine targeting the CTG 18.1 disease-causing mutation, delivered as a first-of-a-kind small molecule pharmaceutical intervention to slow or stop the progression of Fuchs. Thank you very much.